In this short presentation, I'm going to briefly describe the development of an integrated approach to testing and assessment, or IATA, for grouping high aspect ratio nanomaterials within the GRACIOS framework. The GRACIOS framework aims to enable the practical application of grouping, leading to read-across and classification of nanoforms in order to streamline the risk assessment process. Grouping decisions will be underpinned by scientifically justified hypotheses, which identify descriptors relevant to grouping similar nanoforms for a specific endpoint. Within the GRACIOS project, we have developed a number of predefined hypotheses for grouping and read-across. Based on the basic information inputted into the framework by the user, a predefined hypothesis may be suggested as suitable for grouping the nanoforms under investigation. Each predefined hypothesis is supported by a tailored, integrated approach to testing and assessment, or IATA, that will guide the gathering of relevant information, allowing the applicability of the grouping hypothesis to be assessed. The IATA ensures that testing is conducted in a targeted manner that allows integration of existing information with new data generated where necessary, supporting the user in accepting or rejecting the grouping hypothesis. Here I will briefly introduce one of the GRACIOS predefined hypotheses, which focuses on grouping nanoforms based on their potential to pose an inhalation hazard. The specific example focuses on high aspect ratio nanoforms, or HARN, for which knowledge of fiber toxicology, in particular the understanding of asbestos-related disease, helps to inform the hazard endpoints. So following inhalation exposure, asbestos fibers can elicit adverse health impacts in the lung, including mesothelioma, a tumor that arises from the mesothelial cells lining the chest wall and lungs. As the aim of this hypothesis is to group similar HARN on their potential to pose an inhalation hazard, in particular related to the development of mesothelioma, the hypothesis focuses on occupational exposure scenarios, which are likely to result in high inhalation exposure situations. The asbestos literature has generated a structure-activity relationship, which defines the physicochemical characteristics governing the pathogenicity of the fiber, and this is known as the fiber pathogenicity paradigm. Therefore, when we consider what they are for our grouping hypothesis, we define the relevant threshold dimensions, for example, length and diameter, for a HARN to meet the fiber pathogenicity paradigm criteria. These characteristics can also determine where they go, specifically where the HARN will deposit in the lung and whether it can be effectively cleared from the lung and the pleural cavity. Furthermore, what they are and where they go together address the biopersistence of the HARN and the likelihood for their significant retention in the target tissue. What they do frames a similarity assessment between putative group members in the activation of relevant biological responses triggered by known pathogenic fibers leading to disease. The use of the GRACIOS hypothesis template allows a structured hypothesis to be generated, defining clear boundaries of the group and integrating the necessary considerations that may impact a grouping decision. Whether a nanoform or set of nanoforms can be grouped based on this hypothesis can be tested by a tailored IATA. This takes the form of a decision tree, where each decision node asks for specific information related to the key criteria required for grouping similar nanoforms as a HARN with the potential to pose a mesothelioma hazard. Each decision node is measurable with defined criteria to ensure a clear yes or no answer, for example, whether a HARN has sufficiently small diameter to allow deposition in the distal lung. A yes answer allows progression through the IATA, whereas no represents a point of departure where your nanoform may deviate from, and therefore you may reject the grouping hypothesis. Rejection of the grouping hypothesis may prompt the user to consider an alternative hypothesis. For example, lack of biopersistence may lead you to consider grouping hypothesis for nanoforms with quick or partial dissolution. The decision node may be defined by a single characteristic, such as length or rigidity, or a combination of endpoints which contribute to an overall effect. For example, frustrated phagocytosis, which may require a battery of tests or assays to answer. The IATA can support the inclusion of a HARN into a group, and from here, a similarity assessment between group members can support the grouping decision that members of this group may pose a similar hazard, and this can go on to form the basis of a read-across argument. The information required at each decision node can be gathered at different tiers of testing, from simple acellular characterization and in vitro screening assays, to more complex in vitro models such as co-culture or microtissue models, and finally in vivo models including OECD test guidelines. This tiered approach from simple to complex testing can be used to support a grouping decision with increasing levels of confidence. The choice of tier at which you exit the IATA reflects the initial purpose for grouping and the associated level of uncertainty considered acceptable for the user's needs. Therefore, the tiered testing will reflect the level of confidence you require in the substantiation of your grouping hypothesis. We propose a number of purposes for which the HARN hypothesis and IATA will be appropriate, and highlight some of the potential implications resulting from grouping for each of these purposes. So grouping by this hypothesis may facilitate the generation of a read-across argument for risk assessment, and being placed in a group may support the selection of an appropriate source material. Read-across to source materials for which more information is available for the endpoint of interest may allow waiving of testing, for example, lengthy carcinogenicity studies to confirm or reject the mesothelioma hazard. Grouping may also be used to promote the adoption of precautionary measures for materials on which limited hazard data is available, for example, when setting occupational exposure levels or hygiene controls based on other members of the group. This grouping hypothesis may guide and support the innovation of safer by design materials. The implication of adhering to a hypothesis that causally links a specific physicochemical characteristic to an adverse effect may lead the user to alter the particular characteristic linked to the hazard potential. Finally, this IATA may be used to fill data gaps and increase scientific understanding of key mechanisms of fibropathogenicity. This may lead to a refinement of the boundaries of the group, thereby further streamlining the risk assessment process in the future. Here I have demonstrated the practical application of a gracious IATA supporting an evidence-based grouping hypothesis for HARN in relation to their structural similarity to pathogenic asbestos fibers and their associated potential to pose an inhalation hazard. This example illustrates how the mechanistic links between physicochemical characteristics such as size and shape and factors such as biopersistence and toxicokinetics can be integrated to form a well-substantiated grouping hypothesis. For further information on the gracious framework and examples of the gracious IATA, please see our additional video presentations. Thank you for listening.